Hello, good afternoon. We are Team 7 and we will be presenting on the topic of transgenic animals. Introduction. About this unit. This unit was developed by practicing teachers and biotechnology educators from various European countries supported the European Commission's DG12. Its goal is to teach students how transgenic animals are produced and used, while also exploring ethical, economic and social uses related to biotechnology. Okay, this unit is divided into three main case studies, each of which can be used separately or combined. Number 1. A mouse against cancer illustrates transgenic animals as these models focusing on a genetically modified mouse that helps study brain tumors. Number 2. Sumo salmon addresses growth enhancement through a role-play debate about genetically modified salmon. Number 3. Transgenic sheep discuss using sheep to produce human pharmaceutical proteins, A1 proteins inhibitor in milk. The material emphasizes that social and ethical dimensions should be considered alongside scientific concepts, making it suitable for both science and non-science teachers and students. For centuries, humans have bred animals through selective breeding, combining genes only with the same species. Transgenesis, however, allows scientists to transfer genes between completely unrelated organisms because DNA uses a universal genetic code. This technology enables the creation of animals with traits that would be impossible through traditional breeding, such as disease resistance of our medical applications. Examples. The text highlights several famous examples from the Rosalind Institute, Scotland. Number 1. Tracery, transgenic, producing human A1 protein inhibitor in milk. Number 2. Vegan Imora, cloned, the first animals cloned from embryo cells. Number 3. Dolly, cloned, the first mammal cloned from an adult cell. Number 4. Polly, cloned y transgenic, containing a human gene for blood clotting factor 9. Benefits and risks. Potential benefits. High specificity, target traits only. Number 2. Rapid results in the generation. Number 3. Flexibility, cross-space gene transfer. Number 5. Economic advantage, less feed and medication. Potential risks. Number 1. Possible disruption of normal gene expression. Number 2. Virus transmission, especially for organ donors. Number 3. Uncontrolled gene spread into wild populations. Methods of transgenesis, Part 1. The most common method for producing genetically modified animals is pronuclear microinjection. This involves injecting 200 or 300 copies of a foreign gene into a fertility stage and implanting it into a surrogate mother. Only a small percentage of offspring became truly transgenic and even fewer expressed the gene at usual levels. Controls a transgene combinase. Number 1. A promoter sequence controls where and when the gene is active. Number 2. The gene of interest provides the desired trait or protein. Number 3. A poly A tail ensures correct gene function. The promoter sequence is often replaced to that the gene works specifically into target tissue. For example, mammary glands for milk production. Methods of transgenesis Part 2. Other transgenesis techniques include number 1. Retroviral vectors. Number 2. Embryonic stem cell transfer. Another key method is nuclear transfer cloning, where the nucleus of an unfertilized egg is replaced by the nucleus of a donor cell. This method allows creation of genetically identical animals and predetermined sets. Applications of transgenesis. Number 1. These models, transgenic mice carrying human genes help study and treat diseases like cancer. Number 2. Improving livestock, producing fast-grown, linear or disease-resistant farm animals. Number 3. Molecular farming, using animals such as sheep or goats or bioreactors to produce bulbous human proteins in milk.
A mouse against cancer. The biotechnology company Anygen, located in Manchester, had been one of the most successful enterprises in gene technology applied to medicine. It specialized in research on hereditary disease, focusing on transforming bacteria to produce proteins using treatments for genetic disorders such as certain forms of diabetes. For about a year, the company achieved great success selling medicines produced by these transgenic bacteria. However, increased global competition caused a decline in profits, forcing management to lay off a quarter of the workforce and seek new innovation approaches to maintain investor confidence and avoid closure. Research purpose. During a crisis meeting, the researchers decided to change their focus from bacteria to mammals, as bacteria were not ideal for studying biochemical and psychological mechanisms or the genetic basis of human hereditary disease. Since human experimentation was impossible, they chose to use mice as model organisms. Their new goal was to create a transgenic mouse carrying a human brain cancer gene known as BRCA1. This specific cancer was highly aggressive, painful, and affected people of all ages, including teenagers. There was no cure, making the creation of this model vital for researchers into tumor development and drug testing. Scientific background. Transgenic animals are created by introducing foreign DNA into animal cells, allowing scientists to add or deactivate genes. This technology is used to study gene function and hereditary disease. For example, cows can be engineered to produce human proteins in their milk. The most common method used in creating transgenic mice is microinjection, where purified DNA is injected into a fertilized egg per nucleus. Only 15 to 30% of the offspring became truly transgenic, carrying the new DNA in every cell. This method, thought inefficient, revolutionized genetics by making it possible to study disease at the molecular level. Ethical considerations. After initial approval from management, the company policy required that any genetic modification of mammals be reviewed by an ethical commission. This led to the formation of a group including researchers, managers, and ethics experts to evaluate the moral implications of using animals as disease models. The case has become not only a scientific challenge, but also an ethical one, raising questions about how far humanity should go in altering animal genetics for human benefit.
Summon salmon are genetically modified salmon designed to grow much larger and after than normal salmon. They are named after Japanese summon wrestlers because of their size. In 1994, Canadian geneticists and the Vancouver Fisheries and Ocean Department, working with researchers from the USA and Singapore, created a line of high-end transgenic salmon. By 1 year old, The salmon were on average 11 times larger than normal salmon on the same age, and one even reached 13.7 times the normal growth rate. Researchers injecting a growth hormone ranging from salmon into 3,000 fertilized eggs used developed meat having flexibility. After on in only about 6 pounds to present on barbing how salmon expressing the transgenic and salmon impressing growth. However, the transgenic is not fully neutral. Placing the transgenic randomly can disrupt other strains on cause embryos. Only salmon lay eggs on gravel arms in cold streams. Males fertilize them externally summon salmon mantle after or steam capable of breeding and pressing the growth time they offspring. Says anything or working on externalizing summon salmon to prevent the transgenic from spreading to world population. Traps grow large since more salmon stem or consume things from the food industry. Their flesh appears suitable from low calorie randomly main dishes. Mushroom trend in consumer herb and convenience food. In developing mushroom production have viscous increased protein availability and help cable malnutrition. In Sumon salmon escape from fish farms the cute this term inconsistent and reduce biodiversity. They could potentially transmit the transgene to only salmon population. People may rethink genetically modified salmon, especially in labeling is unclear, and saying other transgenic animals hormone imbalances or an unknown infertility could working.
Transgenic prevent the problem of pulmonary emphysema. Pulmonary emphysema is a disease in which the lungs are damaged, causing serious breathing difficulties. It mainly affects chronic smokers, but it can also occur in people with a genetic predispositions. When we breathe, the air contains germs and bacteria that are removed by macrophages, cells that clean the lungs. During this process, macrophages release an enzyme called elastase, which destroys damaged tissues to eliminate bacteria. However, if this enzyme is not cleaned, it can also damage healthy lung tissues. The gene for alpha-1 antitrypsin is located on chromosome 14, and its main job is to protect the lungs. When there is not enough alpha-PL, the risk of emphysema increases, but these conditions can be prevented or treated with alpha 1 PL injections. Ways to get alpha 1 antitrypsin from human blood, which is present and limited by producing in transgenic animals like sheep that make the protein in their milk, processed in transgenic sheep. The human gene that makes alpha 1 PL is injected into sheep embryons. The sheep that are born produce the human protein in their milk. The alpha 1 PL is then taken purine from the milk. After that, it can be used in human patients to prevent or treat emphysema. This means alone large amounts of the protein to be made without harming the animals and gives a safe, efficient biotechnological solutions. Genotypes and population data. These are the three main genotypes. Population data: about 5% of the risk populations is MC, around 2% for half the CT genotypes and produce very little alpha 1 PL. This information shows that a significant part of the population is at risk of lungs diseases if they are exposed to areas like tobacco smoke. Transgenic shift often innovates biotechnological alternative to prevent human alpha 1 antitrypsin, helping to prevent and treat emphysema in people with genetic deficiencies of this protein. Researchers' observations: people with the CT genotype develop lung emphysema much earlier than others. They are more likely to get infections because they have fluid neutrophils. People with the MC genotype, when exposed to tobacco smoke, have higher chance of developing breathing problems. The genic length of alpha-1 antitrypsin combined with environment factors like tobacco smoke greatly increases the risk of developing lung diseases such as emphysema and chronic bronchitis. Solutions and treatment: the lack of alpha-1 antitrypsin can't be prevented or treated with regular injections of this protein. Gene therapy could be a future solution for inserting the human alpha-1 PL gene into liver cells, unless there is a risk of resistant pharmaceutical productions in transgenic sheep. Transgenic sheep produce large amounts of alpha-1 PL in their milk. They are easy and cheap to maintain. The human gene inserting into embryons, implementing into fermenting sheep, and the positive of bringing produce alpha 1 PL in their milk. Treatment of cystic fibrosis and gene therapy. Cystic fibrosis is caused via mutations into gene that makes a protein called CFTR, which controls the flow of cells and water in cells. When this protein doesn't work properly, think mucus builds into lungs and other organs. Gene therapy aims to insert a normal copy of the CFTR gene into lung cells to restore their functions. Although it is still under study, the research are processed as it could improve breathing and patient quality of life. Therapeutic cloning and cell reproduction. Therapeutic cloning aims to augment stem cells indical to the patient to repair damaged organs or tissues. Unlike reproductive cloning, it does not try to create a new living being, but rather to cure diseases. An important example was Dolly, the first sheep cloning for an adult cells. This experiment showed that adult cells contains all the genetic information needed to create a complement organs. Thanks to this discovery, it's become possible to produce compatible organs and develop new genetic treatments.